Good morning, everyone. Today's topic is gram staining, and I'm Uma Maheshwari. First, we'll discuss about the gram staining solutions. Number one, crystal violet can be used or methyl violet can be used. This crystal violet or methyl violet acts as a primary stain. And number two is grams iodine. Number three is acetone or alcohol. This will act as an decolorizer. And number last one is that safranin. It is a dye that acts as a counter stain. Before going further, this gram staining is a differential staining technique. Why it is not a simple stain is that a simple stain will have only one dye. Here we are using two stains. Number one is crystal violet and number two is saffronin. More than one stain if we use, it comes under either special staining technique or differential staining technique. In this case, we tell gram staining to be a differential staining technique because this gram staining will differentiate between gram positive and gram negative bacteria. So we are differentiating two types of bacteria. Hence, gram staining comes under differential staining technique. And the next part is preparation of gram staining solutions. And number one is grams iodine. For this, we need iodine 1 gram, potassium iodide 2 gram. We have to dissolve it in distilled water 100 ml. And number two is counter stain, that is saffronin. Saffronin powder 350 mg can be taken, and this can be dissolved in ethanol 10 ml and distilled water 90 ml. So together it forms 100 ml. Or else we can take saffronin powder for 350 mg and carbol fission for 5 ml and distilled water for 95 ml. Together this also will make 100 ml. So you can take saffronin powder, ethanol and distilled water, or saffronin powder, carbol fission and distilled water. And the next preparation is crystal violet or methyl violet. For this, what we will do is we will take solution A and solution B and mix it together. So solution A is you can take either crystal violet or methyl violet. Along with that, methanol 20 ml must be taken. So this crystal violet 2 gram powder will be taken and it will be dissolved in 20 ml of methanol. This will give you solution A. Solution B is nothing but ammonium oxalate, that is 800 mg. This 800 mg of ammonium oxalate will be dissolved in 80 ml of distilled water. This will give you solution B. Now what we need to is we need to mix solution A and solution B, and you have to leave it aside for overnight. Then next day morning what you have to do is you have to take a Wattman number 1 filter paper or any other filter paper of your choice. You can transfer this to that. You can filter this mixture of solution A and solution B, and you can transfer this filtrate to a bottle where you can be used, where it can be stored for a long period of time. And here comes the procedure of gram staining. So it is a very simple procedure. Number one, you have to take a slide, make a smear of the sample which you want to observe under a microscope. You have to heat fix the smear, and now it is ready. You have to keep the slide on a stand in a wash basin. Then you have to keep it ready. Why we are keeping it over the sink is that, the stain which you pour might overflow sometimes. So in order to avoid making your table nasty, you have to keep it somewhere like over the wash basin we used to keep in our labs. You have to keep it in a slide stand. And now after heat fixing it, you have kept it in a slide stand. Now it is ready to perform. Add crystal violet or methyl violet over that slide on the place of the smear. Leave it for 1 minute. After 1 minute, you take the slide. You have to gently wash it using a tap water. After this, you can add grams iodine and let it stand for a minute. You have to keep the slide on a slide stand, add grams iodine and leave it for a minute. Then again take it out, wash it off with water. Now tilt this slide to 45 degree and add acetone or alcohol drop by drop till you wash the violet color completely from the slide. And care must be taken that you must not add acetone directly over the smear. You must add it on the top of the slide. You will make smear in the middle of the slide. Here you will tilt the slide to 45-degree angle and you will add the acetone to the topmost corner of the slide. And the next step is, once the all the violet crystal stains are moved out, you can wash it out, gently wash it out with running tap water, then add saffron in and let it stand for half a minute. That is enough. Now again wash it off with water. Now air dry the smear. You should not heat fix, you must air dry the slide. After you air dried it, now it is ready to be viewed under microscope. You can apply at first you can see it under 10x and 45x. Later you can add oil and observe it under 100x objective. So in 100x objective it will be much clean and clear. And here is how your observation will be. So I have indicated the gram positive in violet color and I have written the gram negative in pink color to make you realize that gram positive organisms will appear violet in color and gram negative organisms will appear pink in color. So this is the concept. What was the last dye which you added? It is saffron. Saffron is pink in color. So obviously your background will be pink in color. Yes. So gram positive organisms will appear violet in the pink background, whereas gram negative organisms will appear pink in the pink background. And here I have given some of the examples of gram positive cocci and bacilli. At the same time, I have given some gram negative cocci and bacilli as an example. So the examples of gram positive organisms are Staphylococcus, Streptococcus, Clostridium, Lactobacillus. Gram negative organisms are Neisseria, Escherichia coli, Vibrio, and Salmonella. And here is the important part. Most of your Viva questions will come from this part. Here I have included the role of reagents. So primary stain is crystal violet or methyl violet. What is the purpose of primary stain? That is what we will discuss here. So primary stain will initially stain all the organisms that are present in your smear, and it will also stain the background. And later you will wash it down, then you will add grams iodine. Yes, so this grams iodine acts as a mordant, which means this mordant grams iodine will form a complex with the primary stain which you have added already. So it forms a complex, diiodine complex. What this complex do is, it will make the dye to fix firmly to the cell. That is the function of this mordant. And the next one is decolorizer. Acetone or alcohol can be used as a decolorizer. This decolorizer will remove the primary stain which you added initially. This decolorizer will remove the stain of the background. But since the gram negative bacteria is weak, it has very thin layer of peptidoglycan, but it has a thick layer of lipopolysaccharide. Yes. So this lipopolysaccharide will be dissolved and big spores will be formed. Through this big spores, the stains like crystal violet or methyl blue will be removed. So gram-negative bacteria will lose its primary stain, whereas gram-positive bacteria will retain its stain due to the strong diiodine complex that was formed. And this diiodine complex which was formed in the gram-positive bacteria makes the pores of the bacterial cell wall to shrink. In case of gram-negative bacteria, the spores will enlarge and it will lose its stain. In case of gram-positive bacteria, the pores which are already present in the bacterial cell will shrink. This shrinking of pores and expanding of pores is all due to the grams iodine mordant which you add. That will form a complex with the primary stain and do all these things. And the last one is safranin or carbylfuscin. So what is the function of this is that, it acts as a counter stain, that is secondary stain. So this will stain the background and gram-negative bacteria now. Why it is not staining the gram-positive bacteria is that, gram-positive bacteria is already stained with crystal violet. And moreover, the pores which are present in the bacteria is being shrinked. Yes. So again, new stain cannot enter into that bacterial cell wall. So the pores of the gram-positive bacteria are already shrinked, and moreover, it has already formed diiodine complex with crystal violet and grams iodine. So it is now looking at violet color, and this gram negative bacteria is free with big spores. So when you add safranin, it will immediately go inside the bacteria and it will stain the gram negative bacteria, and obviously background will also be stained. So that's why when you look at the observation, all the gram positive bacteria will retain the primary stain and all the gram negative bacteria will retain the secondary stain. This is due to the difference in their composition of the cell wall. So their cell wall composition plays a major role in determining whether a bacteria is gram positive or gram negative, whether it is taking out the primary stain or secondary stain. Thank you.